Good afternoon, everyone. Good morning and good afternoon, everyone. Thank you very much for being here. It's a pleasure for me to open this season number 3, session 7 of our Mixed Gen series, today dedicated to computational methods in biophysics for application to drug discovery. I would like to thank very much our speakers, Romy Amaro from the University of California, Giulia Pagliardi from University of Heidelberg, and Mattia Bernetti from the Italian Institute of Technology. Thank you very much to all our speakers. Just a couple of very quick technical information about the Q&A. There will be 3 seminars, 3 lectures, one from Romy and the other 2 from Mattia and Giulia. At the end of the presentations, you may ask questions. There are 2 ways, basically. You can write your question in the Q&A session, and I will ask the question for you, to our speakers. Or you can simply write, I have a question, and then you can switch your camera and your mic on, and you can ask directly your question to the speakers. Without any further delay, I thank again our speaker, and the first lecture is by Romy Amaro, in situ dynamics reveal unseen vulnerabilities of viral glycoproteins. Please, Romy, the stage is yours.
Thank you, Andrea. Okay, great. Well, it's a pleasure to meet with everyone virtually today for this, I think, what should be a fun CCAM event. Today, my name is Romy Amaro, and I would like to talk to you about our effort to model with ever-increasing realism what we like to call the in situ dynamics of different viral glycoproteins. And so I'll focus today's talk. I'll start out giving sort of a brief description of what we've been doing with SARS-CoV-2, but then I'll also talk about our work with influenza and ultimately vaccine and immunogen design. Okay, so the work in my group primarily involves the application and development of methods to explore the dynamics of biomolecules in scenes. What we're particularly interested to do is to not only simulate the biomolecules, but to do so in a way that really sort of reflects their environment actually in biology as closely as possible. And so we like to refer to this technique sort of as in situ computational microscopy, because we can really now build these models and simulate their dynamics in ways that are getting very close to reflecting their actual environment within the cells. So what we can do is actually take multiple different types of structural data sets taken at different resolution. This could and often include high resolution X-ray crystallography combined together with lower resolution single particle cryo-EM data. And then for our larger scale scenes, we can sort of nest that data within tomography data taken from either Fib milling experiments or otherwise. And then once we have this sort of structural assembly, then we can also include information from glycomics, lipidomics, as well as genomics. And so, we can now build these fairly accurate 3-dimensional physical models of biological systems. And then I think as many of you know, for molecular dynamics simulations, essentially all we're doing is approximating that system down to its many atoms, where we treat each atom as a hard sphere. And then we define this potential function I'm showing here, which basically describes the interactions of each atom with all the other atoms in the system. And then all we're doing essentially is integrating Newton's equation of motion over time. And so we start with a particular structural configuration that has been determined ideally by experimental data sets. I'll talk about that as closely as possible. And then we integrate one time step, we get another structure. We integrate another time step, we get another structure. We do that in numerical integration millions and trillions of times. And we build up this dynamical landscape that then can inform us about, you know, what the system behavior. So I just will mention, you know, nowadays, this computational microscope, as we like to refer to it, is increasingly powerful. And for the simulations I'll tell you about today, we were able to use the PAC Frontera machine, which is at the University of Texas in the United States. I'll change to a laser pointer, as well as the Summit supercomputer, which is at Oak Ridge National Lab. And so what I hope to convey today is how these data-centric simulations are not only being used to integrate experimental data sets, but also to extend them together with new experiments to give, you know, novel insights into several different things, including the mechanisms of how these viral proteins actually function. And that will be the first part of my talk. And then in the second part, I'll talk about sort of related to mechanism, but how these larger scale simulations are actually revealing cryptic epitopes, which you know, haven't been seen before in these in static experimental structures. And then finally, I'll talk about our work to use really sort of advanced simulation methods to accelerate vaccine design and immunogen design. Looking by now that I think everyone is probably, unfortunately, too familiar with the SARS-CoV-2 virus. This is one of, this is a, I don't know, I can't get it to play here because of this. Go back to, well, that's okay. It's not gonna play. That's all right. Anyway, this is the SARS-CoV-2 virus. It looks like a golf ball with spikes sticking out of it. These spikes are actually called the spike proteins, and they, you know, they're an important part of the virus because they sit on the outside of it. So they're the first point of contact that the virus has with human cells. So there's a lot of folks, you know, interested to study the dynamics and function of these biomolecules. On top of that, of course, it's also a major component of the vaccines because they're highly immunogenic molecules. So, you know, understanding the exposure of different immunogenic regions, as well as the mechanism of this protein, you know, is something that's been, you know, of great interest to us and others. So what we've been doing is trying to, you know, build, as I mentioned, sort of computational models that can integrate the existing experimental data, but then also augment this data with modeled, with modeled parts. And so, you know, some of the first data sets that became available, the first one was of the spike protein, was from the group of Jason McClellan, and they were able to resolve at high resolution the ectodomain of the spike protein. The spike protein, like many of these class 1 fusion proteins, are basically, it's a trimeric protein with threefold symmetry. They're fairly, so up here is sort of the end-to-end sequence from 1 to about 1,300 residues. And the part here shown in boldface up to about, you know, 1,140 in terms of amino acids is what they were able to resolve in these cryo-EM experiments. Importantly, Jason's data set gave us a view of the what they call the 1-up RBD, so where the spike protein has one of its receptor binding domains in the upper open position. You can see that here colored in green, and then in the bottom, another terrific structural biology group, the group of David Biesler, use symmetry from the cryo-EM to create a model of the all down or closed system. So we took those two early structures, which unfortunately had a number of missing loops and truncations, but we were able to use Modeler in order to model these missing loops and sort of, you know, fix different disulfide bonds and titratable groups and create these sort of complete models of the active domain. But then beyond just the head portion of the spike, there's also a stalk and a transmembrane component, which are not able to be resolved by experiment, which we were able to model using, at the time we were using IPASSR. You know, nowadays, this was actually before AlphaFold was published, but the models really, they don't change with AlphaFold. On top of the stalk and the transmembrane tail, there's also this disordered bit here called the cytoplasmic tail, shown in purple, that sort of fits inside the virus. And so we can build this sort of end-to-end system that represents the entirety of the, of the like glycoprotein. So the other thing that, where simulations have an enormous contribution to play is in the understanding of the structures and dynamics of glycan. And you'll hear about that in my talk today. I'll focus on N and O-linked glycans of the spike protein and the influenza hemagglutinin and neuraminidase protein. But I think in the second talk you're going to hear more about heparan sulfate-like molecules. So like these glycans are really, I think, becoming more tractable with computation. And I think here we have a nice example of where simulation has really augmented our experimental understanding. So one, a challenge is that structural biologists can, they can see the protein, but because the glycans, these N and O-linked glycans, which at times can be, sort of long and varied in their composition, they're very, as you're about to see, they're very flexible. And so this flexibility, generally prevents the structural biologist from being able to get high resolution structures. And so we can model, so thanks to glycomics information, for example, this wonderful paper from Max Crispin, and there's a sister paper from Paris Suizade, that basically describe the molecular composition or the molecular recipe of these N and O-linked glycans at all the different sites of these glycoproteins. And you can see here for the SARS-CoV-2 spike protein that there's a number of glycans, it's basically covered in glycans all up and down, all along its sequence. And so again, we can model that. But not only can we, build the structural models, but then of course, what we're really interested to do is explore their dynamics. And so what you're looking at right now is actually, the fully built model of the spike protein with all of the glycans there embedded in a lipid membrane that reflects the viral membrane as close as we think it, in terms of its lipidomics as closely as we can manage. And then we're simulating, what you're looking at is the simulation of the atomic level dynamics coming from the molecular dynamics simulation. And so we've run both in the open and in that one open state as well as the closed state. These systems have about 1.5 to 2 million atoms. We were at the time using the Charm 36 force field, running NAMD2 on Taxon Terra. You can see our benchmarks. And we were able to gather some nanoseconds of data in relatively quick order. So, what did we learn from this? Well, of course, we were able to show what the spike protein actually looked like with its full glycan shield, right? And so on the left is really what the experimentalists mainly see, which is the protein itself. And on the right is what that exact same spike protein looks like, but with all of the N and O-linked glycans actually represented. And so here, each of these dark blue tufts represents a composite image of multiple snapshots taken from a 1 microsecond trajectory of the spike protein with the glycans. And what you can see is how each one of these glycans basically sweeps out in effective space, like a volume of space, around the surface of the SARS-CoV-2 spike protein. So then, when they're all together, and they're sort of moving like this, they create this really sort of dynamic coat that shields the spike protein from the molecules of the human immune system. And so this is a very clever way that viruses have evolved to, to mask, to sort of protect themselves when they're inside the body from attack by the immune system. So the other interesting thing about these studies was that beyond just showing what the shielding looked like, which at the time was, of course, of great interest to folks, we also showed how there were 2 glycans in particular that were at positions 234 and 165, that when we rebuilt their glycan side chain under the RBD, the 1 RBD up position, what we saw is that those side chains actually filled up the void space that was created when the RBD went into the up conformation. And so when we were looking at these trajectories, what we realized was that these 2 glycans actually appeared to be acting sort of like a kickstand on a bicycle. And we did mutations in silico, and then Jason McClelland did mutations actually experimentally in the wet lab. And we showed that, in fact, if one deletes these two glycans through mutation, then this domain actually collapses down, and the spike protein is less able to interact with its host cell receptor ACE2. So what was really neat about this work was that it established for the first time a structural role for glycans in the actual infection mechanism itself, which hadn't yet been described for glycans for any biomolecule. So beyond, so what I told you about really in that story was just looking at sort of the relatively short timescales of microseconds dynamics around two experimentally resolved states, the open state here and the closed state. But, there's what we're really interested to do is actually understand sort of a more detailed view or an atomic level view of, oh, am I going to be able to get this to play again? Oh no, I want these movies to repeat. I want to see if I can. Yeah, okay. So I have to get rid of my laser pointer so I can use the pointer to replay the movie. What we'd really like to do is actually to view these molecules, these molecular machines actually moving almost in real time. And so, to do this, we partnered with Lillian Chong at the University of Pittsburgh, and we ran her weighted ensemble method. This is a method that's been developed by Dan Zuckerman and Lillian Chong with help from the community. And with weighted ensemble, it's pretty neat. It's unbiased. We're not adding external forces to the system, but we are sort of following. It's like a statistical method that basically follows. You define a particular progress coordinate. In this case, we defined a two-dimensional progress coordinate that included sort of an RMSD of the RBD going into the up conformation, as well as an angle relative to the center of the spike. And then we're basically running many simulations at once and following the trajectories that are sort of progressing along this opening coordinate. When you do that, you are able to generate many unbiased continuous pathways of, in this case, the spike opening. And so here you can see we're starting from the closed conformation, and then it's going to spontaneously move into the open state. And we can do that with many orders of magnitude less computational resources. So we were able to generate these opening pathways in about 3 weeks on these GPU clusters using this weighted ensemble method, which was really neat. And the other thing I would say about this method is that the efficiency scales exponentially with the energy barrier. So it's it's a pretty nice way to get these views of, in this case, we're interested in the spike in the SARS-CoV-2 glycoprotein, but of course this is applicable to any other number of systems. And so what was to me so curious about this study was that when we found a new glycan, which I'm highlighting here in hot pink, which when we did the original sort of conventional simulations around the two experimental endpoints, what we saw, if you looked at the one up state that I showed you earlier, and you just look at the dynamics, this glycan at position 343 in hot pink, it just looks like in the upstate, it just is sort of sitting on the side of the spike glycoprotein. It doesn't look like it's doing anything interesting. Similarly, in the all closed state, you see that pink glycan at 343, and it's just sitting down at the side of the spike protein. Again, doesn't look like it's doing anything interesting, which is why in those first studies, we basically ignored it. But when we actually did the work to look at the opening mechanism using these weighted ensemble trajectories, what we saw was that this glycan here at position 343 actually participated in helping to, at first, it's sort of interacting. It's going to play one more time and I'll talk you through it. At first, it's interacting with these different residues in the receptor binding domain, and it's forming relatively, tight interactions. Then it starts to basically do almost like a hand jive motion, and ultimately then the RBD, it actually helps to sort of kick it open into the open conformation. So Jason McClellan, again, together with folks in his group, did more experiments, and they showed that if you delete this particular mutation, you have a dramatic reduction in the ability of the SARS-CoV-2 spike protein to open, sort of confirming again the importance of this particular residue. And this glycan, in fact, if you delete it, the virus actually has trouble even getting into cells at all, and it's just an interesting finding. And so, so that was another sort of, I think, good example of where these sort of, again, sort of more these advanced or accelerated simulations using, in this case, weighted ensemble, could inform on the mechanism of glycoproteins. And something else that I think we're particularly proud of is that these simulations have really, I think, taken off. Not just by my group, but as you're going to hear about in the group of Rebecca Wade and many others. I think glycobiology, computational glycobiology and glycochemistry is really reaching sort of a very exciting point where it really has the ability, together with experiment, to provide deep insight into structure and function relationship in biomolecules. And so we were really pleased that our spike protein made the cover of the Bible, as they say, for the textbook of Essentials of Glycobiology, which is really neat. And there's tons of opportunity in the space for folks who, are interested to, who are looking for good projects. There's certainly a lot to do. Okay, so in the second part of the talk, I wanted to focus on our work actually on studying a different virus and tell you about how these simulations actually can inform on cryptic epitopes. So, for many years, gosh, I started when I was a postdoc, which was a long time ago. I have always had, sort of a deep interest in the glycoproteins of influenza, and influenza actually has two glycoproteins. It has on its membrane surface, it has a hemagglutinin molecule, which modulates entry, generally thought of as modulating entry. This molecule interacts with sialic acid receptors in the host cell through this region here at the top, also called the receptor binding domain. And there's been many, many, many structural data sets provided, even by X-ray crystallography, and now, of course, with cryo-EM, that give us high resolution views into what this protein looks like. We and many others have completed those models, missing loops and so forth, adding glycans, et cetera, putting them into membranes to understand their dynamics just in these single glycoprotein simulations. Its sister protein, the HA sister protein, which is called neuraminidase, which is shown here, actually helps to, it's basically like a sialic acid lawnmower, and it comes through and it basically cleaves. It binds the terminal sialic acid inside of its active site, and then it cleaves that last linkage. Okay, and so what that helps to do is to facilitate the destruction or the reduction of the interaction of the sialic acids with hemagglutinin. So it's this delicate balance between binding of sialic acid, which is performed by the hemagglutinin, and cleavage of the sialic acid, which is performed by neuraminidase, that actually affords influenza its fitness inside, in vivo. So in any case, we had not only studied hemagglutinin, but we were also studying this neuraminidase, which sits on a long stalk, which is kind of similar in many ways to the SARS-CoV-2 spike protein. But at the top here, there's a tetrameric ectodomain here. So this is a tetramer protein, and as I mentioned, it is an enzyme. So in any case, we and others had studied for years what the dynamics of these systems were like using these single protein simulations, and we could make all sorts of insights about cryptic binding sites inside of these ligand cavities, which could be useful for drug design. But so one of the things, as I mentioned, that have been really interested to do is to move from, to really not just move ourselves, but hopefully the whole field, to move from studying these glycoproteins, the single glycoproteins in isolation, where we're basically looking at sort of the protein in dilute solutions of water, some buffer, but to actually instead to contextualize these glycoproteins within their realistic biological environments. This data, we now have beautiful data sets this is a rather old one, which is also nice, from this is a cryo-electron tomography data set at about 5.5 nanometers resolution, which gives us a map, essentially, of the glycoproteins on the surface of the virus. This data was taken from the National Institutes of Health from the group of Alistair Stephen, and it gives us, again, sort of this low resolution puzzle in some sense, and we can then fit in higher resolution pieces to these different locations. So it took us about 5 years or so, thanks to the work of Jacob Durant and Lane Vatopka. They were able to sort of convert this cryo-EM data into almost like a point map that where we could trace the viral envelope. And you can see it's not a perfect sphere. I mean, it's fairly spherical, but it does have some shape to it. So we get a, you basically can trace the membrane itself, and then also you can locate the different glycoproteins and then insert into these different locations the high resolution glycoproteins. And you can even tell here by looking at this map, you can differentiate between the two different types of glycoproteins. So the one that looks more like a peanut is actually hemagglutinin, and the one that looks more squarish is neuraminidase. And so we were able to reconstruct this an atomic level model of the entire influenza virion, which had about 160 million atoms when one included water. And I should also say, I mean, I guess I didn't mention this for the first talk, or for the first part of the talk. All of these simulations, once we build them, these are explicitly solvated simulations. So I don't show the waters, of course, because you can't see anything when I do, but the water is actually represented explicitly. So we started with those initial models and then extended them to actually include glycans. And here you can see the glycans actually in yellow. And so we built two different strains. We built the 2009 H1N1 strain, which is shown here on the left. And then we built also the next strain in 2015. And so one of the interesting features of influenza and of many viral glycoproteins, of course, they mutate, as we know. For SARS-CoV-2, I think we know this too well because we've gone, from the original circulating strain to Delta, to Omicron, and, have sort of suffered through this repeat reinfections. For influenza, one of the main ways that it mutates, it actually every 5 to 6 years, these circulating strains, it actually picks up another glycan, usually in the hemagglutinin domain, which basically prevents antibodies essentially from binding. So it in some sense neutralizes the ability of these vaccines to be effective. And then they have to reformulate the vaccine. So we built both of these strains. And then we looked at the dynamics. And so, so this is a view now of the entire influenza virus. And here I'm showing you just a little snapshot of the molecular dynamic simulations, the trajectories that were run. And I'm trying to call your attention to this blue molecule here in the middle. And when you watch it, I hope you see something that actually one of these domains appears to be sort of opening up. There it's closing again, but there seems to be like plasticity between the actual HA trimer itself. And so another view of this, just focusing the view on a single glycoprotein. So this is that same hemagglutinin, but here looking from the side and here from the top. What you'll start to see is that this hemagglutinin is actually breathing open. And here you can see this chain as it goes further out, we color it more yellow, but you can see that it does actually, you saw it it starts here very tightly packed. And this tightly packed configuration is, of course, what most of the structures, almost all the ones that we've seen that have been resolved from cryo-EM at high resolution, they're always in these tightly packed states. But we very quickly saw dynamics of this open. And so I should also mention, because I didn't have it in the slide, that we were, for the whole virus simulations of about 160 million atoms, we had replicas of about 500 nanoseconds of dynamics. So, we're not, because of the system size and computational resources, we're not able to run out to, tens or hundreds of microseconds, which would be ideal if we could. but we're instead limited to shorter time scales. But even with these short time scales, we're seeing these sort of interesting motions. The other thing that we can do with these large scale simulations is that because there are so many copies of each of the different glycoproteins in the scene that we're simulating, right? So I'll go back for influenza, we have 200 and some copies of the hemagglutinin, and we have 30 or 50 or 60 copies of the neuraminidase shown in red. And so what we can do is actually we can use Markov state model theory to essentially deconstruct the whole virus into its constituent pieces, and then actually extract longer timescale dynamics from many short time scale simulations, which we can sort of discretize into, or we can discretize the molecules into different states and then compute the transition between the states. What we've done here for this HA in particular is we've been able to sort of quantify the breathing motion. So one way, of course, you can quantify it is just by looking at sort of a histogram and looking at the head-to-head distance between the different RBD trimers. We can actually look at the head-to-head distance. And so we can see that there's the dotted line is the crystal structures that we started with, and the cryo-EM structures we started with. But you can see there's quite a range around that exhibits different opening states. And then we can also quantify the breathing motion itself. And so, as the HA RBD sort of goes into the, breathes open and closed, this is happening on the order of sort of hundreds of nanoseconds time scale. So just another view of this opening motion, if we looked at the RBD at the distance between that one of the receptor binding domains and the core of the protein, this is now we're looking at a time series analysis over the 500 nanoseconds that we sampled from one particular of the hundreds of HA molecules. And here you can see the three different chains, this distance, this pink line here. So we start when they're all tightly packed. Again, that's representative of the structural data that we get from, from experiments. This line here is showing what an exemplar sort of tightly packed structure looks like. We refer to this as closed. Over time, as you saw, it's like undergoing different motions. By the time it gets to about 300 nanoseconds or so, this molecule actually breathes open, and you can see here one of these open states. And this is now a super open state, as you can also see there. So what was interesting about this was that it turns out in 2020 or so, the group of Ian Wilson and James Crow and others actually published a study of a new antibody called Flu A20, which they had found that actually indicated that it was binding to a site on the hemagglutinin trimer that was considered mostly to be buried if you looked at, when they looked at all the structural data sets of HA. And so, I just want to read here from this paper. We were sort of really thrilled to see this because it indicated to us that this breathing motion that we saw may actually was likely relevant for the binding of this antibody. And I'll just read this little paragraph here, which is taken directly from their paper. It says, therefore, the HA molecules on the cellular or viral surface generally have been considered to be stable trimers, with the trimer interface regarded as inaccessible and thus not targetable by the immune response or therapeutic. And so I just want to pause there, because this is really, I think, the key point is that we have this enormous ability now using technique, particularly with cryo-EM, where we can get these very high resolution structures. But, the focus in structural biology is always driving towards the highest resolution state. And we drive towards this state really at the sacrifice of understanding the dynamics of these molecules. And so, it's more than meets the eye with all of the molecules that we know from X-ray crystallography and cryo-electron microscopy at this point. And simulation is, obviously a key tool in our, sort of in our scientific strategy to understand what really are the dynamics in vivo that these molecules are undergoing. But okay, little sidebar. Just continuing on, I think the really important part is the next few sentences where it says, the ability of flu A20, which is that antibody, to confer in vivo protection strongly suggests that HA molecules are dynamic and more heterogeneous in their conformations than we have observed previously, and that the trimer interface is partially or transiently accessible. And that's exactly what we sampled in these large-scale mesoscale simulations of the HA. And again, we were able to model the flu A20 just to show you what that looks like. Here's the flu A20 bound in an attempted binding to the closed state. You can see it totally has steric conflicts, clash, but in the open state, it's actually, this antibody can in fact be accommodated. Then I just, I want to tell you about another finding actually for the neuraminidase molecule. So the neuraminidase molecule is shown in red. Here we're like going back to the view of the whole virus and the molecular dynamics simulation. And what we saw here, which was really striking, you can see how this neuraminidase head is actually tilting. It's really tilting sort of over to the side. And it's interacting with the different glycoproteins that it's sort of, that are in its neighborhood. And so here's another view of the NA head tilting. You can really see, so this is a stalk, ordinarily, I always tended to think of the NA molecule as being sort of, as just sitting atop the head, but here you can sort of just evenly sort of orthogonal to the stalk, but here you can see it's like really, really tilted. And so, we thought this was pretty curious. And actually, I was giving a talk at the NIH Vaccine Research Center, and I wasn't even talking at that point about neuraminidase because we weren't really sure what to do with that data. Instead, I just showed this movie of the full virus, and Masaru Kanyukiyo, who's a fantastic structural biologist there, actually saw this motion, and after the talk asked me about it. I said, well, what I saw was really interesting. Okay, so I think I can probably skip forward to this, but so anyway, so we actually can characterize, of course, the head tilt angle as well as the kinetics, similar to what we did with hemagglutinin. But what we did when I was talking with Masaru, it was very interesting because it turns out that he had data that he had not yet disclosed, he and his group, where they had discovered what they were calling a dark side neuraminidase antibody that was extracted from infected patients a patient who had been infected with H2N2, so this convalescent plasma. And so, so here what I'm showing is data that he showed us. We have here in the middle in red. So this is negative stain data. You can see in red here is the neuraminidase tetramer. In orange is an antibody that they know, this is called the 1GO1 Fab antibody, that they know binds to an epitope that's on the top side of neuraminidase. So neuraminidase also has, epitopes that are immunogenic, and we don't put them generally in the vaccines for various reasons. But that binding site of epitopes on the top side of NA has been known for a long time. And you can see here how it occupies very clearly the density on this top face of neuraminidase. But what they also saw in those same data sets was very clear density of a Fab that they were calling this neuraminidase dark side or NDS antibody that they that was new. And so what they were really excited about, one of the things that they were always curious about was how this epitope was actually accessed when the full stalk was there. And so so what our simulation showed was how the neuraminidase head could basically tilt relative to the stalk, exposing this epitope that could in fact be bound by this dark side this dark side antibody. And so the two structural biologists who were really sort of leading the data acquisition on this is Julia Lederhofer, and then, as I mentioned, Masaru Kanyukiyo, who's the group leader there at the NIH Vaccine Research Center. And this image here just shows how it just basically, it's a predicted model of a Fab antibody bound into the envelope of data that they see for for this dark side antibody. And this was published maybe in October of November of last year. Okay, so in the final part of the talk, I wanted to just briefly tell you about our work to use simulation, not only to understand mechanism and cryptic epitopes, but also to actually be relevant for immunogen or vaccine design. And so S2, so now I'm going back to the SARS-CoV-2 spike protein. And, as many of us know, this whole, we have the vaccine, and it's, it's sort of less and less effective, of course, as the strains, mutate and circulate. But then we would get like another vaccine that's been encoded with yet a different version of the spike protein that includes both the S1 domain, which is shown. This is the full spike ectodomain, which I told you about earlier. Most of the vaccines are, they're encoding the whole thing, but most of the epitopes that we think are neutralizing are in this S1 domain, which sort of sits on top of the S2 domain. So the S2 domain is actually the S2 is sort of it represents sort of more of the interior part of the spike protein, where the S1s basically come off of it. And this is actually exposed during the process of viral fusion. Of course, the S1s do come off, and this whole central helices basically polymerize upwards and interact with the host cell membrane and facilitate viral fusion, of course. But S2 is curious because it's also among the most conserved parts of all of the spike proteins across the different beta coronavirus family. So it's long been considered one of the leading possibilities for potentially developing a broadly neutralizing vaccine. And of course, this would be good because if we could deliver or develop and then deliver a protective neutralizing vaccine that would handle all of the different variants as well as other coronaviruses, that would of course be great from a medicinal standpoint. But it's various challenges. So we set up together with Jason McClellan and a great team of immunologists to try to design S2 derived immunogens. Okay. All right. So in order to do this, something I sort of glossed over before was that the spike protein, it is highly dynamic. As I showed you just a glimpse of sort of the short time scale dynamics, around the different states. But in order to get the high resolution structure, in order to work with this protein and deliver effective vaccines, they're not just delivering, in most instances, the vaccine doesn't contain just like the circulating sort of wild type sequence. They actually are using a mutated construct that is easier to work with experimentally, but also that in the case of the spike protein freezes the conformational landscape. Actually, it biases it into that one-up conformation. And in fact, that's how Jason McClellan, together with his colleagues, that's how they were able to achieve the high resolution structure, is that they had these 2p mutated, they call them 2p mutated structures, because right at the tip of this, at the central helices, they have proline mutations that stabilize the protein and that again bias the conformation to the one-up state. That one-up state, of course, has really important, it's good for the characterization of the high resolution structure, but it's also that it exposes that key immunogen, which is that receptor binding motif that interacts with ACE2. And that's one of the reasons why those early vaccines were so darn effective, because your body was presented, in fact, with a bias, with this one open state that had this epitope just exposed. In any case, when you're doing immunogen design, you really need to work with the construct that they're actually working with experimentally. So we so the constructs that we were working with include the original 2P mutations, which they disclosed a long time ago, which were actually taken from originally from discoveries in the MERS glycoprotein. But now then subsequent to that, I think it was like in May of 2020 or by June of 2020, Jason had also continued to work to stabilize further the spike construct, and they developed something called the Hexapro variant, or the Hexapro construct, which is, I think, the one most widely used now in labs across the world. And the citation is here. And that has an increased thermal stability of about 5 degrees Celsius. It also has really much improved expression yield, and it maintains this pre-fusion state in post-thaw, which is again really important for structural studies and so forth. So this Hexapro has an additional 4 proline mutations here. So in all told, it's a mutated sort of base construct, and that's now the dynamic that we're interested to work from as we try to do design of sort of like the next possible immunogen that would use this particular construct. So the issue was, I should tell you that the issue was that this S2 state had been even stabilized with this Hexapro, with all of these proline mutations. It was never stable. It was still very conformationally diverse. Okay, so it's still very flexible. Without the S1 domain on top of it, this S2 portion would basically splay open, and they were not able to, even for example, they were not able to achieve a high resolution structure because there was too much conformational variability. So we set up to try to stabilize using, as I'll show you the methods that we went out to use. Jason and his groups and other experimentalists had been trying to, sort of like guess which mutations they could introduce that would stabilize this construct. Our tack was, again, to go back to weighted ensemble simulations. And so we just started simulating just the S2 portion. So what I'm showing here in purple, this is just the inside part of the spike protein now with S1 gone. And we used conventional, nothing really interesting happened in the conventional simulations other than it just sort of started to fall apart. But we repeated the weighted ensemble method on this particular construct with the thinking that if we could understand the opening mechanism of this S2 construct, that we could then potentially strategically mutate the residues that were involved in that opening mechanism in order to develop an S2 construct that would, where the opening would be prevented or, slowed. So we did all these weighted ensemble simulations similar to what we did before. This gives us, many of these opening pathways. And then we look at the mechanisms in terms of what we saw. So now I'm sort of, in order to sort of convey what the opening mechanism was, here, what we found was that there were 3 sets of helices at the interprotomer interfaces that really mediate the opening. So here, this is like a cartoon schematic of the S2. Again, it still has the trimeric symmetry. You can see it here. And it has, there's sort of there's 3 helices per chain that are interacting, across these different interprotomer interfaces. Okay, and so we're representing them here. And what we saw after looking at all these opening pathways, what we saw was that it's sort of the opening mechanism. What happened was at first, one of the chains sort of starts to break away, and you can see sort of a loss of contact. Then this chain also loses its other contact here and sort of breaks away. And then eventually these other 2 chains separate. And so based on this mechanism, what we saw was actually, I don't think I show it here, but it actually was sort of a zipper. It opened by almost also like a zipper-like mechanism. And so once we understood those particular residues, this actually, we came to an additional set of mutations. This what we call these interface mutations, and I'm not disclosing them yet because we haven't published publish this. We're getting very close to putting in the archive, but I'm sort of hiding the exact residues right now that we were able to mutate. But they actually created, so based on our simulations, they actually created experimental constructs. And what they found was that the mutations that we introduced, which were derived from the opening mechanisms that were deduced from weighted ensemble, basically stabilized, even further stabilized the S2 construct. So it had even more superior thermal stability, which was terrific. It had even better expression yields, which was also very good. And this double mutant, one of the mutants that we made, the so-called interface double mutant, was stable enough that they were able to, at long last, actually resolve the high resolution cryo-electron structure of just S2, which is shown here. So then subsequent to that, this has been, put into mice and so forth, and they're doing a big set of neutralization assays and so forth, which again, I don't include here, but will be disclosed hopefully sometime this summer. So that's sort of the end of my talk. I guess there's sort of a lot of people to acknowledge. I have a really terrific group out here at the University of California, San Diego, in La Jolla. We've had a sort of sad stretch of not sunny weather, but ordinarily it's sort of a little paradise over here in the southern western coast of the United States. And of course, I have lots of folks in my group to thank, including Lorenzo Castellino, who really led really the opening domain work, and also the spike work that I showed you with the glycans, although that was also a huge team effort. Christian Seitz did all the Markov state model analysis and really helped tremendously, again, with the sort of whole virus simulations, as well as Sarah Kahanek doing some early work there on our earlier studies. Also, our experimental collaborators, Julia, Masaru, and Ian, lots of folks for funding. I also want to thank Jason for his, or Jacob, who's now at the University of Pittsburgh. He has his own wonderful lab there. He's somebody who was really brave, I would say. Something that these very large simulations are in a lot of ways high risk. They take a long time to make. They take a long time to simulate. You never know what you're going to get. And it's really hard work. And he was someone who, from the very beginning, was just really fearless in just doing it. And since then, he sort of helped blaze a trail that was continued by Lorenzo and by Abby and others, and Abby Domer here, who's also really a fantastic big system builder. So I just have to give them extra thanks. And then also our theoretical colleagues, who helped with the application of the Markov state model theory, as well as, I guess, missing from this. Oh my goodness, yeah,
Okay.
missing from this, is Lillian Chong. Okay, yeah, happy for questions.
Thank you very much, Romy. Very,
No problem.
very interesting, wonderful presentation, actually. And now it's time for questions. And as I said, you may either write your question in the Q&A and I will ask question for you, or you can simply say, I have a question, and you can switch your camera and mic on and directly interact with Romy. So there is one question from Obakim. Would you like to ask your question personally? Should I do for you? Would you like to switch your camera on? Probably not. So Romy, Obakim is asking how many MD simulation samples did you collect for building your Markov state models?
Okay, right. So in this case, we had we ran the full virus simulations in triplicate. And so there were 3 500 nanosecond replicas of those. But then each of those, had, like, for example, with the hemagglutinin, molecules, there were 236 hemagglutinins. So there was, many replicas, like hundreds of replicas per virus, and then there's 3 of those. So there's a considerable amount. I don't know how many hundreds of microseconds of sampling in the end when you factor that all out. So there was quite a bit of data that we gathered for the MSMs.
So I'm seeing Joachim is here. So if you want to further
Oh, great. Yeah.
comment on this, Joachim, please go ahead.
Thank you very much. But mainly the issue of Markov state models is collecting enough samples to build a fully Markovian model, because there can be many times that you have cells, okay, dozens of samples, but still the model is not Markovian because there is not the initial, and well, it's quite complicated. I was wondering. So how many samples did you collect to be able to build a fully Markovian model, which passes the Chapman-Kolmogorov test?
Oh, yeah. And so this data I didn't show you here because it's sort of a higher level talk. All of those, convergence metrics and so forth are in the paper, that central science paper. But I mean, we had thousands of simulations of the hemagglutinin separate, that were each about 500 nanoseconds long. So we had hundreds of microseconds of sampling for hemagglutinin, and then we had also hundreds of copies of the neuraminidase. But yeah, I mean, you're right. It is something that one has to be careful with, and it also depends on exactly, how you discretize the states and so on and so forth. I think the methods for, developing Markov state models now, yes, it's still challenging, but I mean, it's much easier now with tools like PyEMMA that have been developed by, for example, the NOE lab. I think they're pretty good. Mm-hmm. But yeah, it's a pain.
Okay.
Thank you very much.
Thank you, Romy. Elena Spinetti, would you like to ask your question?
Hi. Yeah, thank you. Thanks a lot. Great talk. And I was really impressed by the full virion simulation, but my curiosity is, let's say, what's the advantage of having it instead of having like relatively big simulations, but like patches with multiple proteins and doing replicas on these? And like if you have extracted additional information from the virion.
Okay, yes. Oh, so that's an excellent question. So it does seem possible. And in fact, I would agree with you, even though we haven't shown it, certainly for influenza in a rigorous way. I do think that the simulation of crowded patches of glycoproteins can likely yield very similar dynamics about these glycoproteins. The closest we come to that was a really nice paper from Gerhard Humer with the SARS-CoV-2 spike protein, where he and his team built a small patch of, I believe it was 3 or 4 spike proteins sort of crowded together. And when they did that, they did see sort of larger scale, interdomain motions of the spike protein, particular the ectodomain relative to the stalk. And they could also see the stalk bending as the spike protein was interacting with its neighboring proteins in this smaller patch. So I think, yes, I do think that we can break up these models, into smaller patches, which does make them more computationally accessible. I think the exact truncation that people use is not, I think that's still a bit of an art, I guess I'll say. I don't think we have rigorously calculated well, how big do you actually have to simulate, how crowded does it have to be in order to replicate the dynamics? That's not been done, but it's something that could be envisioned. And then, but I would also say, so for, you have to, so for us too, I mean, I think, well, why didn't we do that to begin with? Well, in part because we wanted to, my ultimate dream, I think that many of our ultimate dreams is to, we want to simulate the whole virus and actually its interaction with cells. And so, with host cell surfaces of the lung and so forth. So in order to do that, it seems like, one step towards that is build the virus, the whole virus, where we have where we can do that based off of good experimental data, right? This cryo-electron microscopy, where we can get all of the crowding and the density correct, and we don't have to make decisions about truncation. So there's always like a trade-off in that. That's, so I think that's one of the advantages, of just including the whole system. You don't have to make that choice. And then, we're working on, I guess I would say, subsequent studies are looking at these different features that are sort of at the whole virus level. I don't have anything to share with you there yet, unfortunately.
Yeah, I understand. Thank you. Very cool. Thanks.
Thank you, Elena. Giancarlo, Francese, I'm seeing you
Yes.
would like to ask a question.
Yes, very nice talk. Thank you. My question is about the explicit water simulation. You were mentioning that you're doing everything in explicit water, but is it fully atomistic, or are you using coarse graining and then coupling in some way with water?
It's fully atomistic. It's pretty brute force.
So I mean, that means that you're using, several millions of water molecules, right?
So much water. I will tell you, Giancarlo, we have so much water. And the first thing we do when we are analyzing these systems, because they're so big, is we strip all the water out. And we don't throw it away, but we just focus on the protein dynamic. So there's all of this water, which we keep, which we feel we must keep to get the dynamics correct of the proteins and the virus, but then we haven't analyzed. And if anybody wants at that data, we're happy to share it with them. I mean, that's one of the other things, I guess I left this out of this particular talk, but that I sometimes mention is that these, especially the big simulations, there's so many possible questions. There's so many different pieces of this puzzle to look at, from lipid interactions, water interactions, and I think that there's potentially great value in just sharing this data openly so that people can go in and ask whatever questions they want for this data set, because we certainly can't go in and answer them all ourselves.
Well, that's a very good point. I mean, that definitely something that we could do, try to analyze together the data. That's a nice thing to do. So thank you for your answer.
Sure.
Thank you, Giancarlo. Isabel, please ask your question.
Yeah, hi Romy, thanks for the nice talk. Hey. I was wondering how sensitive your large-scale simulations are to force field issues, as for example, for charm, it's known that carbohydrate protein interactions turn to aggregate more than in experiments. I mean, it turns out that your simulations you showed are quite successful and reproduce experimental findings, but do you also have unsuccessful systems?
Okay, great question. So I have to say that I want to make a separate comment on that, and that is for the spike glycoprotein simulations. We partnered with Elisa Fada, who's an amazing glycobiologist / glycochemist at the University of Maine, and she ran almost the exact same simulations, but with amber, the amber force field. And that's a funny question that just got asked. And what we saw was that the dynamics were the same. So I think that it appears that when we're looking at sort of these questions about dynamics and so forth, I think both the glycan force field and the charm force field, so the amber and charm force fields, appear to be yielding similar results. So I think that's great. But then, beyond that, in terms of stickiness, in terms of free energy, of binding and this sort of thing, we haven't really done, I guess I can't really say, we haven't done a very careful analysis of comparing different force fields. So I would be a little bit hesitant to try to address that. But I guess I would say that, in these cases, even with the charm force field, which maybe is a bit more sticky, as you mentioned, we've been able to replicate experimental data quite well.
Okay, cool. Thanks.
Yeah.
Thank you, Isabel, and thank you, Romy, for your reply. Is there any other question? You have a general curiosity, Romy.
Yeah.
With these pretty large simulations, pretty long simulations, which were, apart from being trivial, only 7-8 years. So probably are not trivial even now, but still at least doable. We test the numerical stability of MDN genes, which was something not, again, super trivial, because we are not solving analytically the Newton equation of motion, but we have some numerical tricks. And nobody knew 10 years ago whether these tricks were stable over multi-microsecond and for multi-million or even billion particles.
Yeah.
And also, as already mentioned, also the force field stability, because force fields are empirical and therefore can be stable for a certain amount of simulation time for certain system, but less stable for other systems. And also force field stability and force field reliability has been addressed by these super big simulations and super long simulations. Based on your experience, which is rather remarkable in the field, which are the next challenges from the technological standpoint and from the physical standpoint? We need to run faster simulations. We need different codes. We need to envision something completely new because we reached the upper limit for parallelization. Which is your perception? I know that there is no unique answer, so it's more brainstorming, but I would like to know your opinion and probably also to stimulate some thinking for the younger attendees.
That's a really great question. You know, I think that you know, that there obviously still are force field deficiencies. You know, there are challenges there. I think that, I guess for me, sort of what I reflect on being sort of the biggest challenges are really improved force fields that are, you know, good enough to actually reliably determine binding free energies, you know, not just in boutique, bespoke ways where people are, you know, but in sort of more broadly reliable ways. And that's a whole field of study, you know, and I think work there has certainly the potential to impact the work at the larger scales also. And then the other thing that I guess, you know, I am very excited about the continued development of enhanced sampling methods and our ability to predict and develop better ways to explore the dynamics of these complex systems, you know, more quickly. I think that's also great. And then the other thing that I guess I would say is I think multi-scaling and doing more efficient multi-scaling where one can really sort of switch the physics to, you know, efficiently and again, reliably, sort of, you know, because at each scale, whether if you're at the very small scales, you know, worried about something like a very detailed calculation where you're interested in very large scale, you know, ultimately, you know, we want to, I think the dream for many of us is to really have a computational microscope that can inform from the molecular to whole organ or, you know, even whole organism scales, right? Sort of really a digital twin of what's happening. But in order to do that, we have to, you know, for these extremely large scale simulations, we really don't need to carry around all of the behavior of every single atom, right? And so an improved ability to sort of switch between scales, to exchange the physics, to answer and ask different questions of these different systems, I think it's to me one of the areas which I think is really the grand challenges, and there's many people working on that. So I think that's great. I think it's a bright future.
Thank you very much. Many ideas for future projects for younger investigators in particular. Thank you, Romy. Thank you very much again. And
thank you.
Now we leave the stage to Giulia Paiardi from Heidelberg University with presentation entitled The Role of Heparin in Spike SARS-CoV-2 Infection from a Model for Heparin Sulfates to a Starting Structure for Antivirals. Please, Giulia.
Okay, can you hear me and can you see my screen?
Perfect.
Okay, perfect. Not easy to talk about viral glycoproteins after Romi's great talk as always, but I will do my best. I will thank you for inviting me and for giving me the possibility to tell you what we are doing in Rebecca's weight group about the SARS-CoV-2 infection, and in particular, what we are studying for the spike and heparin. Okay, can you also see my pointer here? Okay, I can imagine, yes. So
yes.
just to briefly refresh to you the mechanism of infection of SARS-CoV-2. Here, the SARS-CoV-2 is depicted as a cartoon with the spike shown as blue sticks. So the first encounter between the SARS glycoprotein and the host cell receptor is mediated by the interaction of the spike, here depicted in S1 and S2 portion, with the ACE2. Thanks to this interaction, the SARS-CoV-2 can exploit two host protease, furin and TMP RSS2, which cleave the spike in position S1, S2, and S2 prime. So letting this S1 portion to detach, activate the S2, and so allow the fusion with the host cell membrane. Since the main actor of this story is the spike glycoprotein, I will give you a brief description about it. So the spike glycoprotein is an homotrimer. So it's composed by 3 of the sequences reported here that in the structure are shown in different grades of gray. So here I also reported the spike in 2 different conformations, as Romy already mentioned, in the closed conformation and in the open conformation. The main difference between these 2 conformations is the exposition of the receptor binding domain, because in the closed conformation, the receptor binding domain is not exposed to the solvent, is shielded. So it cannot bind the ACE2, while in the open conformation, the RBD is exposed to the solvent, so it can bind the ACE2, so in this setup here. The last point I will mention in all this story in the sequence is that I reported these blue boxes here, which are reported also in the structure here. And these blue spots are called basic domain or heparin binding domain, and are composed by basic residues which electrostatically interact with polyanionic sugar chain, as we will see later, or any anionic charge protein, let's say. So just to go back to the SARS-CoV-2 infection here, if you see the presentation of the membrane, it seems like that the ACE2 is a tree in the middle of the desert. But if we want to really depict the surface as a cartoon, we need to show that the ACE2 is embedded in a glycocalyx, and it's surrounded by a lot of glycoproteins. So why are those important? Because it has been experimentally demonstrated that the heparan sulfate proteoglycans facilitate the SARS-CoV-2 infection. And what are these heparan sulfate proteoglycans? Nothing more than proteoglycans, so core protein, on which are covalently attached this heparan sulfate, composed by repetition of this disaccharide unit, which is variably sulfated. But it has also been demonstrated that the heparin acts as an antiviral, preventing the binding to the heparan sulfate. And as you can see again here, heparan sulfate and heparin share the same disaccharide, and for some region also the same sulfation pattern. So a lot of time, heparin is also used experimentally as model for the heparan sulfate proteoglycans. And here, let me raise the question we have, because we know a lot about the biology of this mechanism, but we are missing the mechanistic information about it. So what we want to address, the question we want to address are mainly 3. How does heparin exert its antiviral effect, and how do the heparan sulfate proteoglycans facilitate this infection? And the idea is to use these 2 answer and try to answer these two questions to then optimize new heparin analog acting as an antiviral. So let's move on the first question. In this case, I will try to be quite fast because of the time limit and because we already published these results. So by performing over 20 microsecond all atom ND simulation of the spike in the closed and in an open conformation with 0, 1 or multiple heparin chain, we were able to characterize the antiviral effect of heparin. Something I didn't state yet, but it's important to notice is that even in our case, the spike glycoprotein is simulated fully glycosylated, so with all the 66 N-glycans attached to the spike F2 domain. So in this case, as I said, by performing the simulation, we first of all show that the heparin is able to bind either the closed spike or the open spike, spanning from the basic domain of the receptor binding domain till the basic domain in the S1, S2, either in closed and open conformation. And in case of the closed conformation, heparin is able to hinder the activation of the spike, while in the open conformation here, heparin is able to allosterically affect the end region responsible for the fluctuation of the receptor binding domain. But what is really interesting is that based on our model, the heparin is not able to shield the receptor binding motifs, so the specific residues that interact of spike RBD, which interact with the ACE2. So based on the structural similarity between heparin and heparan sulfate proteoglycans, we also suggested that even the heparan sulfate proteoglycans can similarly bind to the spike glycoprotein, facilitating the encounter between the spike and the ACE2. And this allowed me to move on the second question. How do heparan sulfate proteoglycans facilitate the SARS-CoV-2 infection? Obviously, explain to you, just introduce in fact that our spike model with the open spike were not shielding the receptor binding motif. So what we did was then to superimpose the ACE2 to our spike heparin system, and we build the open spike with the ACE2 bound and the open spike with the ACE2 and the heparin bound. And from this, we perform over 10 microsecond molecular dynamic simulation of these two systems here, comparing them to obtain new information about the mechanism of action. So first of all, we focus on the interface between the spike RBD and the ACE2. So at first, we evaluate the residues interaction, because as I mentioned, what we did was to superimpose the crystal structure of the ACE2 onto the receptor binding domain. And this analysis allows us to identify that during our simulation, 90% of the interaction were conserved along the MD, either with ACE2 bound or with ACE2 and heparin bound. The only difference between these two systems was the ones reported here. Indeed, in the system with ACE2 and heparin bound, the interaction between tyrosine 41 and asparagine 501 was disrupted in presence of heparin. And for who maybe know more about the spike, this asparagine here is a hotspot of mutation within the spike receptor binding domain. What we next did was to perform a cross-correlation matrix to evaluate if the heparin was able to somehow affect the movement of these residues here. And as you can see from the cross-correlation matrix on the right, we have the value for the system in absence of heparin on the top and in presence of heparin on the bottom. And obviously, it's really easy to see and to notice that in this portion here in particular, heparin is able to promote the cross-correlation movement of these residues. But which are the corresponding region within the interface? And interesting, those are far from each other, because in case of the spike RBD, they represent the loop free, while in case of the ACE2 RBD, They represent the distal alpha helix residues here. And this data also correlate with a more rigidification of these two regions only in presence of ACE2 and not upon ACE2 bind. Sorry, so only in presence of heparin and ACE2 and not upon ACE2. So after this, we also noticed in our simulation that the ACE2 was bending toward the N-terminal domain of the spike toward the SC spike. So we define and we characterize this movement by using multiple values. So first of all, we define the distance between the center of mass of the ACE2 and the N-terminal domain. And then we also retrieve and compute along the simulation the amplitude of the angle alpha and beta, which are defined by the z-axis and two vectors, the vector alpha and beta. In case of vector alpha, it goes from the center of mass of the receptor binding domain to the center of mass of the ACE2, while in case of beta, it goes from the center of mass of the alpha helices till the center of mass of the N-terminal domain. And here are the results. In orange, the values for the system upon ACE2 and heparin binding, while in blue, upon only ACE2 binding. As you can see here, in case of heparin and ACE2 binding, the distance between the two center of mass decreases, while the variability of amplitude for angle alpha and beta is decreased again upon ACE2 binding, suggesting that in absence of heparin, only upon ACE2 binding, ACE2 tend to have a swinging motion, which is also reported in cryo-IM structure, while in presence of heparin, this orientation is more controlled, is more oriented. And last, this trend was also confirmed by the eigenvector of the first principal component for the simulation in absence of heparin, so only upon ACE2 binding, the movement of ACE2 here and the movement of the N-terminal domain here are pointing towards two different directions, while in case of heparin binding, the movement of these two K regions are pointing toward each other, suggesting once again, confirming once again that the heparin is able to orient this movement. So next, something we again noticed was this buffering effect of the N-glycans that also Romy already pointed out, the role of N-glycan. So what we did was to characterize the interaction of these N-glycans by performing H-bond analysis. So first of all, we identified a buffering effect between ACE2 and spike and terminal domain in principle, which is driven by the ACE2 and glycan 546 and 322, together with the interaction of N-glycans of spike 17, 165, and 343. Something we also noticed was a direct interaction between ACE2 553 directly together with heparin. And once again, also this handshaking between the spike and the ACE2 driven and promoted by the N-glycan N19 of ACE2, which is also reported in literature. Obviously, even in absence of heparin, all these interactions are maintained with the exception of the interaction between N53 and heparin. But since these N-glycans seem to have a kind of a gluing effect, we also investigate their role by performing Tau-RAND, random acceleration molecular dynamics simulation, which is a specific technique implemented by a former postdoc in Rebecca's group, Daria Koch. With the Tau-RAND, what we do is to give a randomly oriented force to a ligand, or in this case to ACE2, to facilitate, to promote the dissociation between ACE2 and spike. And this allows us to compute the relative residence time. And by performing this analysis, what we obtained was the following. So the presence of heparin was increasing the relative residence time. However, since the affinity between ACE2 and spike is really high, we need to use huge forces. So for this reason, we are more interested in the mechanism of dissociation more than in these real values. So what we obtain and why we have these two differences here. So here I reported the egress pathway for the system in presence of heparin. As you can see, we have three metastable states. In the first metastable state, we have the disruption of the buffering and glycans, which I already disrupted by, but the protein-protein interaction here represented it as A is still there, as well as the interaction N53 with heparin. In the second metastable state, the interaction between N53 and heparin is disrupted, and this promotes and unlocks the protein-protein dissociation in which follow a fan-like movement. And in the third metastable state, the protein-protein interaction is disrupted, and the N19, which is the last anchoring point of the interaction, is gliding outside the pocket. What's the difference in absence of heparin? In principle, the metastable 2 is stable because we do not need this unlocking interaction between N53 and heparin. And in the metastable 3, the sliding is faster because the pocket where the glycan is inserted is less narrow compared to when in case of heparin. And with this, I would like to give you a summary about this first point of the presentation and collecting our results. So heparin is possibly the heparan sulfate proteoglycans, because here we are using heparin as a model for the heparan sulfate. It strengthened the spike ACE2 interaction, increasing the cross-correlation movement. And it also orients the ACE2 bending toward the spike and terminal domain. Finally, it concurs in stabilizing the complex by better packing the complex and increasing the residence time of ACE2. We also get new information about the N-glycan, which exert a buffering effect between spike and ACE2. They strengthen the interaction between the two proteins. And unfortunately, I do not have time to discuss about the last 2 points. So, as I said, we have still one point. Can we optimize new heparin analog acting as antiviral? So why do we want to do it? Because in principle, heparin is one of the most potent antiviral identified right now. The problem is that it also has an anticoagulant effect. So we cannot use it really as a first stage inhibitor for the interaction. So our aim is to identify heparin analog with similar antiviral effect, but no anticoagulant effect. And for doing this, we are using a workflow, the following workflow: coupling biochemical screening, mechanistic characterization, VLP inhibition, and inhibition of the infection using real viruses. Obviously, what we are doing is reflected in point 2. So we want to characterize the mechanistic effect of 2 compounds which have been identified by biochemical screen. So at first, we perform 8 microsecond MD simulation of these 2 compounds, compound 1 on the left and compound 2 on the right, on the closed spike glycoprotein. So what we identified and what we notice is that the interaction between heparin and these 2 compounds is very similar compared to the binding path of heparin. We also characterize the central distance of loop 1 on the receptor binding domain to evaluate if this compound were able to better packing the closed spike, and this is the case if you compare it with the open spike and with the open spike with the heparin bound. So what's next? We also perform 10 microsecond MD simulation of the same compound down to the open spike. And what I didn't mention yet is that these conformations are the most representative conformation along the trajectory of our simulation. And as you can see here, the receptor binding domain in compound 1 and for compound 2 are quite different. So since we already identified the end region responsible for this fluctuation, what we did was to perform and to check the deviation of this hinge region to identify if they were really affecting the movement. So as you can see, the movement, the deviation is really comparable to the ones of heparin. So once again, we perform a behavioral principal component analysis on this region. And as you can see, the population identified are quite different compared to our standard and also to our heparin system. So we then perform an essential dynamics to evaluate what was going on in this up receptor. And as you can see, in case of the compound one, it seems that the receptor binding domain rotate. This is the first eigenvector of the principal component. While in case of the compound 2, it seems that the up receptor binding domain is rotate and closing on the spike. So we confirm and we evaluate if this effect was real once again by performing the surface exposed to the solvent of the receptor binding motif. And for the compound 2, it's really clear that this compound is able to shield the surface exposed to the solvent, while in case of compound 2, we are still struggling to understand further mechanisms. And with this, I would like to conclude, giving you final notes about the effect of these two compounds on the closed spike. They seem to be able to induce hinder the activation by binding the receptor binding domain of one monomer and the N-terminal domain of the adjacent one. While on the active spike, they allosterically affect the hinge region responsible for the fluctuation of the receptor binding domain, inducing the closure of the up RBT. So with this, I would like to conclude. Thank you all of you for your attention, and to all the collaborators, in particular to my collaborators in the MCM group.
Thank you, Giulia. Thank you very much. And now is time for questions. Ovakim, please, if you want to ask directly your question, switch your camera and your mic on.
Thank you. Just cannot turn the video, unfortunately. It says you cannot start your video because the host has stopped it. So it's okay. So thank you very much. So the question is, since you're using principal component analysis, inherently with PCA analysis, you lose some information because, so the question is, how many dimensions for PCA did you keep for the analysis?
Okay, in principle, what I did was to compute the principal component for the first 10. And then after doing it, I evaluated that the first principal component analysis was more or less 5 times bigger than the second one. And then we had a scale very similar. So for this reason, we mainly focus on the first principal component. But we obviously check also all the other one. The reason why we focus on the first was the magnitude compared to all the others.
Mm-hmm. Okay, thank you very much.
Thank you.
Thank you, Ovakim. Thank you, Giulia. Other questions? I have a curiosity for you, Giulia.
Mm-hmm.
The way you are targeting the protein is interesting, is rather new, I would say, and may need to be 100% confirm some structural activity relationships to be put in place. Are you planning? Are you envisioning? Do you have already some preliminary data in this direction to build pretty large SAR? I know that one may need a certain amount of compound, but do you have an idea in this direction?
Yeah, I mean, in principle, these compounds were selected from a biochemical screening. In particular, it was done on SPR data, and they chose the 2 which were better able to inhibit the binding to the heparin. And obviously we are doing the computational characterization, so the mechanistic characterization, but together with this, we also have experimental collaborators who are testing these compounds using VLP, so virus-like particle. But as soon as we know that sometimes the virus-like particle, they have a different trend compared to the real viruses. We are also testing the activity of these compounds in inhibition assay using real viruses. While in case of the first part of the talk, so the part related to the ACE2 and the heparan sulfate effect, we are lucky because there is plenty of experimental data already available. So we are not directly testing, but only referring to the literature to cross-correlate and confirm what we see.
Okay, thank you. I see there is another question from Archana Patel. If you want, you can ask directly the question, otherwise I can do that for you. Probably you cannot switch your mic and camera on, so how were the glycans added to the spike protein, Julia?
They were covalently bound, obviously, to the aspargine, and as well as Romy already mentioned, we follow the glycomics profile described by Crispin and the collaborators. So we use, to practically do it, we use Glycam web server.
Okay, and force field parameters are good?
Yeah, we use Amber
adequate
parameters. Yeah. Exactly, as Elisa Fada did in Romy's collaboration. Yeah.
Okay.
Thank you.
Oh, thank you, Archana, for switching your mic on. Is there any other question or comments? Okay, if this is not the case, thank you again, Julia, for being with us today. And now the last speaker is Mattia Bernetti from the Italian Institute of Technology. And Mattia is going to share with us some ideas about RNA, in particular using molecular simulations towards RNA targeting with small molecule drugs. Please, Mattia, the stage is yours.
Okay, can you hear me? And can you see my screen?
Yes, perfectly.
Okay, perfect. Okay, thank you, and good afternoon, everyone. I will first give you a very brief overview of this talk, and so I will first give you a brief introduction on RNA relevance. Then I will show you a practical example on how to reconstruct RNA ensembles, and then how we can use these concepts towards a drug discovery perspective. I will then conclude with a very quick summary. Okay, so RNA historical relevance has traditionally been based on its role as an intermediate in the flow of the genetic information from DNA to the production of functional proteins. However, over the years, we've seen how RNA molecules can actually play many functional roles. And as a result, RNA molecules can also be involved in pathological conditions, opening the possibilities to therapeutic strategies. So aside from vaccines that we heard of and we all know nowadays, basically we can envisage two main strategies. The first one involves devising small molecules able to bind RNA targets and modulate their activity to achieve a therapeutic effect. Another strategy is the possibility of developing RNA oligonucleotides, which are relatively short RNA sequences able to achieve an effect through several mechanisms. What I would like to highlight is that in both cases, but in general, it is essential to achieve a thorough characterization of the involved RNA structures. So first of all, of course, to understand RNA function, but also, for instance, to assist the design of RNA-targeted drugs, or also to understand the structural effects of chemical modifications of RNA oligonucleotides, and so on. But as we know, RNA structure is is challenging to characterize, mainly because RNA molecules have a highly marked structural flexibility, and they also display complex structural dynamics. So for this reason, we typically see RNA molecules as ensembles of conformations more than a single prevalent one. For all these reasons, it is rather difficult to characterize RNA molecules experimentally, and so we can take advantage of computer simulations, in particular of molecular dynamic simulations. We have seen today in the other talks also. So with molecular dynamics or MD, we can simulate the evolution over time of biomolecules at atomistic, with atomistic resolution and at near physiological conditions. So in reconstructing RNA ensembles, MD faces the usual issues that we know. So the first one is related to the accessible timescales that we can try to overcome by using dedicated enhanced sampling methods or using specifically devised collective variables. And the other issue is the underlying physical model that we use to represent the biomolecules in our simulations, which is the force field, basically. So in this case, to overcome these limitations, we can either optimize the parameters, or we can integrate experimental information in the simulations. And this generally can be done in through main ways. The first one is by using the experimental information during the MD simulation, so using the experimental information as restraints, or after the MD simulation in a reweighting way. So I will now show you a practical application of the combination of enhanced sampling methods and reweighting techniques to reconstruct the conformational ensemble of an RNA molecule. Okay, so in this case, the experimental data that we used are small angle X-ray scattering, or SAX data. These experiments give low resolution information, meaning that they do not provide atomistic resolution information. They tell us more about the degree of compactness of the structures. So if you look at the spectra here below, for instance, this is the so-called Kratky representation of a SAX spectrum. We know that a SAX spectrum with a higher peak and a lower tail is typical of more compact conformations, while a lower peak and a higher tail are typical or correspond to more extended conformations of the biomolecules. So another thing nice about this type of experiments is that they are done directly in solution, so they provide us with ensemble information about the biomolecule of interest, and also they are rather robust against varying buffer conditions. In this particular case, the experiment was repeated several times for the same molecule by varying the ionic conditions, as you can see here. And as a result, as you can see from the change of the shape of the SAX spectrum, the molecule under study changed conformation. So these are real experiments, and these were done on the GTPase Associated Center RNA molecule, or GAC RNA, which is a 58 nucleotide long RNA. It is found in the ribosome, and it's involved in protein translation. So the point here is that given these different experimental spectra that we have, we did not know what was the underlying atomistic conformational ensemble corresponding to the different spectra. So to identify these conformational ensembles with atomistic detail, so to achieve this goal, we devised a pipeline where we first generate an heterogeneous ensemble with conformations having different compactness, and we did this with enhanced sampling simulations. Then we computed the SAX spectrum for each one of these MD sampled structures, and then we applied reweighting approaches to find the ensemble that best matched the reference experimental SAX data. So for matter of time, I will show you all the 3 steps very briefly, but I will try to clarify a bit how the pipeline works with a bit more detail. So in the first step, we use the non-set sampling MD simulations. In particular, we use Hamiltonian replica exchange metadynamics. I will not go into the detail of the collective variables, but as you can see, the result of this simulation was to sample structures of the GAC RNA at different compactness. You can see here a more compact structure, a more elongated one. And so once we had these structures, the next step was to compute the SAC spectra for each one of these structures here. So to do this, we can use the Debye relation. Again, without going into detail, I would like to make you note that basically here we have to compute pairwise distances for all of the atoms in the biomolecule. So this means that the computation of a SAC spectrum given a structure becomes quite computationally demanding very easily. So to overcome this limitation, so to improve the efficiency of the computation of the SAC spectra, or in other words, of our forward model, we used an approach that was devised by the group of Professor Carlo Camilloni at the University of Milano, where we basically use we do MD simulations at the typical atomistic scales with buffer and everything. But then when we have to compute the SAC spectra, we resort to a coarse-grained representation of our system. In this case, the pairwise distances are way lower in number, and so the computation is more efficient. And it has been shown that basically you can get equivalently accurate results. Okay, so we have an heterogeneous ensemble of different conformations for this GAC RNA molecule. We have computed the SAC spectra for each one of these structures. Now we want to use reweighting approaches to find the ensemble that best matches the experimental SAC data. So a way to do this, to do reweighting, is to use the maximum entropy principle. Through this approach, we essentially look for the posterior distribution, so for the posterior ensemble, which is as close as possible to the original one, so the MD sampled ensemble, and best matches the experimental observation. So in this case, we selected the 2 most different spectra. So the first one obtained for the RNA molecule in presence of magnesium ions, and the second one obtained in presence of potassium ions that you have here in gray and pink respectively. And as you can see here in green, the spectrum computed from the reweighted ensemble in presence of magnesium matched very well the reference experimental data in gray. Also, the reweighted the spectrum computed from the reweighted ensemble in presence of potassium also was compatible with the reference experimental data in pink within statistical uncertainty. But if we want to get a closer look of how the conformational ensemble looks like, the reweighted ones, if for instance we look at basically the ensemble that reproduced the experimental data obtained in presence of magnesium, we can see that the reweighted ensemble basically had a little fraction of extended conformations. Differently, to reproduce the experimental data obtained in presence of potassium, the fraction of more extended conformations was way more significant. So this work was published in 2021, and all the data to reproduce the results are freely available. There is also a Jupyter notebook to reproduce all of the analysis and all the figures in the paper if you want to play with it. Okay, so the reconstruction of reliable RNA ensembles gains particular relevance if we want to study also the interaction of RNA molecules with biomolecular partners, particularly with small molecule ligands, for instance. So this type of interaction, the target ligand interaction, from a computational standpoint, is typically studied, predicted, say, through docking-based procedures. So these procedures have been quite extensively applied and used for protein targets. However, if we want to extend this application to RNA targets, we have to consider several critical aspects, like for instance, how to include the target flexibility, how to identify druggable pockets, or how to improve dedicated or optimized scoring functions for RNAs. So concerning the first aspect, so how to include the target flexibility, a possibility is to represent the RNA target, the RNA molecule, as an ensemble of conformations. So in this case, we have to repeat the docking exercise for each of the conformations in the ensemble in what is called an ensemble docking fashion. So within this context, we are currently working on the MALAT1 RNA, which has a complex three-dimensional structure that you see here. In particular, this 3D structure includes a triple helix motif that you see highlighted here in color. These RNA molecules involved in different pathological conditions, including various cancers and neurodegenerative diseases. And it has been suggested that the identification of small molecules able to bind and alter the formation of this triple helix can be a viable therapeutic strategy. So there is currently a crystal structure for this RNA molecule in the unbound state, so with no ligands. However, unfortunately, there are no experimental data around. Nevertheless, our plan to tackle this system was still to use molecular dynamic simulations to build an ensemble of a first guess of ensemble of conformations, and then to use docking or virtual screening to identify possible ligands of this RNA molecule. So, so far we have done unbiased MD simulations using, I would say, standard parameters. As you can see from the root mean square deviation with respect to the initial state, the structure was quite stable, but moved a bit far away from the initial conformation, and this was also confirmed by the ERMSD. This parameter here, the ERMSD measures, is specific for nucleic acids and basically computes if the base pairing pattern deviates with respect to a reference. In this case, the reference is the initial conformation, and typically values below 0.8 correspond to structures where the overall base pairing pattern is maintained with respect to the reference. So to analyze with more detail our MD simulations, we used an algorithm named Pocketron that allowed us to identify the pockets of this on this RNA molecule during the MD simulation and identify the most persistent pockets. Then we also performed a pocket crosstalk analysis to identify possible communication patterns between these pockets, and this allowed us to identify two major sites where to possibly study ligand binding. So if we take a look at the focused ERMSD on these two sites, we can see that indeed during the MD simulations there was a certain degree of conformation variability. So what we did is to perform a dimensionality reduction analysis, so principal component analysis on the conformations sampled, and also a cluster analysis, and so and then use the centroids of this cluster analysis as a first guess of an ensemble of representative conformations for this MALAT RNA. So our idea at this point is to actually expand the conformational space of this RNA molecule by using enhanced sampling MD simulations, and then to repeat the analysis and see how much conformation variation we can get, and if we can see something particularly interesting on the triple helix. So with this said, I will give you a quick recap of the presentation. So I discussed very briefly RNA relevance and how prospectively we are moving in the direction of drug discovery in this context. I showed you a practical example, an application on this GAC RNA on how to reconstruct a conformational ensemble by combining MD simulations and experimental data, in particular small angle X-ray scattering data. And finally, I showed you our initial effort on the MALAT1 RNA to reconstruct a conformational ensemble to study in a possibly drug discovery perspective. So with this, I would like to acknowledge all my colleagues and mentors at University of Bologna, IIT, CISA, and also the National Center for Gene Therapy and Drugs based on RNA technology, the project on which I'm working now. SECAM for the very nice invitation to present my work, and CINECA for the computational time. Also, if you want more details, I am also at the poster session, so you can come by and we can chat. We have also another poster from our group, by the PhD student Stefano Bosio. So please go visit and take a look. And thank you for your attention.
Thank you very much, Mattia. Very interesting presentation, and now it's time for questions. Just as a general discussion, Mattia, do you see any overlap between what can be used to simulate RNA and methodologies in the large field of molecular dynamics used for proteins which are not structurally well defined, the so-called intrinsically disordered proteins, or even more small domains or small regions, which are more and more interesting also from the drug discovery standpoint? There is any parallelism between?
So, I would say from a computational standpoint, so very, very practical also, I would say to study, to be able to study these challenging systems such as intrinsically disordered proteins or IDPs, you have to use essentially enhanced sampling simulations. This is so far, I would say, the only way to go to access their highly complex conformational space. So yes, in a way, I would say that more or less the same methods can be applied to challenge RNA molecules, and actually have started to be applied, the same enhanced sampling methods to study RNA molecules, mostly for smaller ones, but also for very recently for larger RNA systems. So yes, I would say, practically speaking, I can see a parallelism computationally wise.
Okay. Even though from the force field standpoint, the situation will be, of course, completely different. Hey, there is one question from Andrew Emerson. Probably, Andrew, if you want, you can switch your camera and mic on.
Hi, hello. Well, it's my colleague Byler who's with me in CINECA. He would like to ask the question.
No, I was just curious. I mean, first of all, it's a nice presentation. Thank you. How did you assume the centroids when you did the clustering? And which method you used for clustering?
So I used QT clustering, which is called the quality threshold. You can take a look. We also there is also some detail in the Jupyter notebook that I shared on how to practically use it. Also because I also used it for the other simulations. So basically, to get the centroid, you get the structure which is as much similar to all the others within the cluster. And for this, I used as a metric the ERMSD. So to do the clustering, I used the G vectors, which is quite a long story, but I will try to make it short. So to compute the ERMSD, you basically use vectors which connect the different bases. So to understand how much the base pairing pattern is similar with respect to a reference. So you can compute these vectors here, and you can use those to do dimensionality reduction, but also for computing a distance between different frames. So using this, basically you compute the ERMSD between all of the structures within a cluster. In general, in the simulation. So you can divide in clusters, and then you can redo the same to find the centroid of the cluster.
So centroid was assumed as one, which is closest to all the other frames in the same cluster, or?
Yeah. Yes, exactly.
Okay, thank you.
Welcome.
Okay, thank you.
Thank you for asking. And also RNA will be a big challenge for a supercomputing center like Cineca, because so far we are simulating very small oligonucleotides. But if you want to go for a real messenger RNA or such big system, we need certainly your help from the physical, technological, and hardware standpoints, of course. Yeah. So glad you are here and you share with us this new challenge in the field of computational biophysics. Are there questions from the attendees, from the participants? If this is not the case, Julia, probably? No. If this is not the case, I would like to thank again.
Sorry, I was
yeah,
not
yeah.
fast enough. I have also curiosity more as a question, because I noticed that you use the polarizable force field for the water, but OPC, if I'm not wrong, and FF99, if I'm not wrong. I've never worked with RNA. Did you test either TIP3, a more classical water model, and also OPC, and did you saw any advantages, or is it something that it's the rule, or maybe you already showed that and I lost it during the presentation?
No, I didn't show this detail. That's actually a very good question. So OPC is a 4P model, essentially. So the straight answer is no, I didn't test the comparison with the TIP3P, but there are, I would say, at this point, several works around that we can consider as a test for using OPC with this, let's call it standard RNA force field, which is the KIOL-OL3, the one that I basically used. So I would say more or less the combination, if you can afford, is now the standard, the suggested way to go.
Okay, thank you. Thank you so much.
Welcome.
Okay. Thank you very much, Mattia, again. I also would like to thank again all the speakers, Romy Amaro, Giulia, and Mattia. And now we have 10 to 15 minutes of break, and then we can meet again in the Gator Town for the poster session. So I look forward to meeting you there. And thank you very much to all the participants, and see you for the next MixGen series. Good afternoon. Bye-bye.